Okay, here we go with part two. We're going to start with a joke. You guys think my jokes are so funny. So the lady's saying to her husband, "You don't look anything like the long-haired skinny kid I married 25 years ago. I need a DNA sample to make sure it's you." Ha ha ha. Yeah, to give your obligatory laugh. All right, moving on. So translation is a second process. This is the one where you're converting from RNA that we created in transcription into a protein. You do not need to write down the things that it has in the box there. Those are just a list of the different amino acids that are necessary for building protein. So there's 20 different amino acids, and we talked about how there's 20 different transfer or tRNA molecules that carry each of those different ones. You have a table of this in your textbook as well, but you definitely do not need to memorize it. The different amino acids all have different chemical makeup, so this chart just kind of shows the different chemical compounds of all the amino acids. If you take any biochemistry classes in university, you'll probably have to memorize what the chemical makeup is of the different ones, but for Bio 30, you do not, so you can be happy about that. Okay, translation. What happens is that the mRNA code that we've copied from the DNA code is read in groups of 3 nucleotides, and we wrote down earlier that that's called a codon. And there are 4 different types of nitrogen bases, and if we're reading 4 different things in a group of 3, that gives us 64 different possibilities of order that those can be in. There is a table in your data booklet, okay, that you should have from when we did pedigrees, and there is also the same table in your textbook on page 670 if you don't have your data table with you. You should be bringing your data table with you though to class. So if you look on that table, you'll see that where you have AUG, actually I'm just going to skip ahead to the table here. So here's the table. This is the one from your data booklet. The one for your textbook is a little bit different, but it works the same way. So I'm just going to move to my mouse here. What you have is when you're looking at your codons, the first base, either U, C, G, or A, is down the side. The second base that is in your codon, you look across the top, and then the third base you use this column on the side. So if I was looking for the sequence ACG, okay, my first base is A, so I'd come to this A column. My second base is C, so I would come to this one, and then that narrows it down to 4 possibilities, and I can find ACG is threonine, for example. So you're going to need to be able to read this table. So if you look, some of them have stars beside them, and there's a little note at the bottom about what those stars mean. So if I go back to this slide, if you haven't finished copying it down, the AUG is a start codon, which is always the amino acid methionine. And there are 3 different codons in your table that say that they are a stop codon, and they actually do not code for an amino acid. Okay, this is just a diagram showing what the different codons, so 3 mRNAs, we have these shapes that are drawn to represent chemical compounds, and we'll get into that a little bit more in a minute. So we have this diagram again, so you should have your table out in front of you. Okay, same 3 steps in translation as in transcription: initiation, elongation, termination. However, something different is happening here. So in initiation, a ribosome is going to actually bond right onto that strand of RNA. If you remember from Science 10, when you do the cell, your ribosomes are found. They just look like tiny little dots floating around either in the cytoplasm or attached to the rough endoplasmic reticulum. And the ribosome can come apart into 2 units, and those 2 units literally clamp down with the strand of mRNA in between the 2 of them. And the ribosome is going to slide along, again, direction matters, in the 5' to 3' direction until it finds a start codon. So until it finds one of those AUGs in a row along the mRNA. Okay, so I'm going to draw you a picture here. Again, you're going to need to draw this down. I'm going to use the same strand of RNA that we created from the picture from transcription. So it looks a little bit like this. A, C, G, A, U. Okay, So what happens is my ribosome clamps down on top of this. Okay, and the ribosomes basically are the size of about 6 amino acids, so that's why I've drawn it that way, with the top part and the bottom part clamp on together on top of it. And then my piece of DNA is going to be read in 3 nitrogen-based sections, which is called a codon. So here's a codon, here's another codon. Okay, and then I have the U at the end, which would be part of the next codon. We have tRNA molecules floating around in the cytoplasm, so they're kind of these big things that look a little bit like a T, and they have amino acids. I'm just going to put an AA for amino acid attached to the bottom of them. So I'm going to draw in a couple of tRNAs. I'm going to draw another one over here. As we all know, I am not the best artist, so bear with me. There's another different amino acid, and I'm going to have this one be UUG. This sequence that is on the tRNA is called an anticodon. Okay, so here I have another anticodon. So the anticodon pairs up with the codon. So what you need to do is, my question is, which one of these tRNAs would attach to where? So AAC, what would AAC be able to pair up with? Okay, so I'm looking at this first codon here. What would AAC be able to pair up with? Normally in DNA, A would pair with T. However, in RNA, I do not have T's. I have what instead? U's. So this tRNA molecule here with UUG will pair up to AAC. This one here, CCU, will pair up with GGA. So in a ribosome, there's enough room for 2 tRNA molecules to attach, and they carry with them at the bottom down here their amino acids. How do you know what amino acid it's carrying? You need to look in your data table. So grab your data tables back out, open them up, and take a look. If you look at the title of this table, it's called Messenger RNA Codons and their corresponding amino acids. So one of the big mistakes people make is that they look up what the anticodon is instead of looking up the codon. So I need to look up the codon. So if I want to do this first codon, the one here, AAC, then I need to look up AAC on my table, not UUG. So when you get out your table, you want to look up A and then C and find out what amino acid that codes for. So you can pause it here, give people a second to look it up. Okay, did you get asparagine? If you got asparagine, you are correct. So you could even write in for AAC, this amino acid here that it's carrying is actually asparagine. What about the other one? So are you looking up GGA or are you looking up CCU? Okay, take a second, find it on your table, and you can pause it if you need to, and then come back and I'll tell you what it is. So you should be looking for glycine. So this tRNA molecule would be carrying glycine with it. Okay, and again, reading in the 5' 3' direction. So once this ribosome is done, it's going to shift on and move along this way. You would be told for Bio30's sake which direction it's going. Okay, I'm going to erase this diagram, so if you still need a minute, make sure you have that all copied down. And moving on to the next slide. So after that, so that's kind of initiation where we have the binding of the ribosome, and now we're going to move on to elongation. So 2 tRNA molecules come into the ribosome and attach side by side. Once they're attached side by side, a peptide bond, which is a kind of bond that forms in a protein, will form in between the 2 amino acids that are being carried by that tRNA molecule. Once the bond has formed, the ribosome is going to shift over one full codon. Okay, so I'm going to draw another picture to show you kind of what this would look like. So we need our same sequence again. So same sequence of DNA that we've been working with here. Or sorry, this is mRNA, not DNA. So it's A, C, G, G, A, and then I had a U, and I'm just going to add in a couple more codons here, another U, and what did I put here? Oh, 3 U's in a row, just so that we have another full codon that we can deal with. And I'm just going to simplify this a little bit. My tRNA that was attached had the anticodon here. Okay, this tRNA had the anticodon CCU. Okay, and what ends up happening now is I have a peptide bond joining between this amino acid used to be attached to this tRNA, but once the peptide bond forms, it detaches and it's bonded and now held on to this one. Okay, my ribosome then used to be attached here. Oh, there we go. My pen wasn't working for a second. So my ribosome used to be attached here. It's going to shift over. Okay, so now I'm going to erase it. You don't need to have drawn this ribosome in. Okay, and you don't need to draw it in over here. But basically the ribosome shifts over one codon. So now it contains this piece with my 2 amino acids, and it has an open spot. And in that open spot, a new tRNA molecule, one that pairs up with UUU, which would be AAA, can fill that spot. It's carrying with it its own amino acid, so you can check on your sheet to see on your data booklet what one would that be carrying. So again, I look up my mRNA codon, not the tRNA anticodon, so you would want to look up with this one UUU, which is phenylalanine. Okay, so it would be carrying phenylalanine here. And then once it has come into place, it's going to form another peptide bond between this chain, and then this will detach. You don't need to write this part. It's hard for me to show you this on a picture. So this will detach. I'm left now with this tRNA that has the long chain floating off of it. My ribosome would then shift over another section. It would just continue along until it reaches one of those stop codons. So that, I'm going to switch it here now, so pause it if you need to, if you're not finished copying that down. Again, don't worry too much about having the ribosome drawn in. Oops. Okay, I switched slide, but I need to erase this, so just give me a second here. There we go. So termination, when a stop codon is reached, so one of those 3 possible stop codons, the whole thing is basically just going to detach. So the ribosome opens up, the mRNA comes apart, the protein chain comes apart, and you are left with a long chain, which then can go through the folding process that proteins do in order to be used. Okay. I liked this one because people always get those nice armband tattoos, so this is just kind of showing what a geneticist might think is a real cool tattoo. You are likely feeling pretty confused right now. Don't worry too much about it. We will be going through this again all day tomorrow if you manage to get this far. You have a worksheet that's going to be handed out that looks like this. So if you can grab that worksheet, I have 1 minute left in my video time. I'm going to try and really quickly go through this. So you have DNA strand number 1. DNA strand number 2 is the complementary strand to strand number 1. So in DNA, A pairs with T, C pairs with G. Okay, the mRNA on the worksheet, it tells you that this is being transcribed from strand number 2. So mRNA, T goes with A, C with G, T with A. The tRNA is the anticodon that would pair up. So A would normally go with T if it was DNA, but there's no T, so it's got to go with U, G, U. And then amino acid, I'm looking up on my table, what would the amino acid be for the mRNA codon, which is ACA, so it's threonine.